I know you've been hearing a lot of information about a lot of different therapies and development. We are fortunate in Duchenne to have 8 FDA-approved drugs for Duchenne, 8 drugs that have been approved in 8 years, and we're very grateful for that. And then you've been hearing about so many more that are in development and in clinical trials, and we're feeling really hopeful about that as well. But the truth is, we have so much more that we need to be doing. And that's what this session is about. This is to give you a little sampling of the type of innovative research that is still going on to do more, to do better, to build upon the advances that are already made and we're already seeing, and to even go beyond so that everyone with Duchenne will have access to safe and effective therapies that will really transform their lives. And this is all new research, and we're going to hear from a number of excellent speakers. But I just want to say that all of this stuff is still at the preclinical stage. So it's newer science than what you've been hearing in the scientific sessions. And this isn't going to be in clinical trials this week or maybe this year, but it's just to give you sort of an idea or a sampling sort of how we're not just resting on where we're at now, and we're really looking forward to how we can keep doing better. So with that, I'd like to invite my first speaker. There you go.
Thank you, Leanna. I'd like to thank Duchenne to invite us here to present our research work. So my name is Ye Liu. I'm the VP of Gene Therapy Research and Regenetics Bio. Yesterday, my colleague Naz has presented our progress for RGX-202, an AAV8-based microdystrophin therapy in clinical trial. Today, I'm going to show you data of one of our early research work on one-time dose AAV-mediated exon skipping in a mouse model for DMD. This is our forward-looking statement. This is my disclosure. So this morning, we have a wonderful session again, moderated by Liana about exon skipping, the basis of exon skipping, so I don't have to spend time to tell you about the background. But very briefly, to tell you in plain English, if you think your DNA as a book, as a message, the wild type dystrophin should be the sentence of you get the big red box. However, if the B and I, the 2 red letters, are missing from the message, then what is it? You got the gibber, it's a gibberish, right? You couldn't understand. So what we are trying to do is to artificially skip the letter G. The message becomes you got a red box. So it's not a complete message, but you got 18–19% of the message right. So currently, there are several antisense products for exon skipping already being approved in the market after accelerated approval. And they normally result relatively low level of dystrophin from 1 to 5%. And the treatment is delivered through weekly infusion. Although this morning we already heard there are new technology that can try to improve from weekly to monthly or bi-monthly. And confirmatory study of this treatment have yet to support clinical benefit. But however, let's say the top 7 exon in the mutation hotspot, we can able to benefit almost half of the all DMD patient. So we know that exon skipping, this technology is working, but it require many frequent treatment, and the main issue is delivery. How do you deliver this effective medicine to the nucleus where it's actually needed? At Regenetics Bio, we are trying to combine exon skipping with AAV gene therapy by delivering a 1-dose exon skipping therapy using something called U7 small nuclear RNA. Well, Dr. Kevin Flanagan tell you a little bit about how to change U7 into an ASO delivery mechanism. In this figure, that's a structure of a U7 small nuclear RNA. It has important function in our cell by modifying the histone mRNA. So researchers, including Dr. Flanagan and us, we have tried to engineer the small nuclear RNA So by changing a small protein binding site, we optimize it so we can go to the nucleus more efficiently. And we also figure out a way to combine 3 different antisense sequences into this U7 structure. Therefore, we can maximize the skipping efficiency. We also package this U7 cassette into an AAV so it can efficiently target the muscle, and we are basically changing the patient's muscle cell into a biofactory so it can produce its own antisense oligo to do the exon skipping. So we are currently in early research phase for both exon 51 and 53 skipping. So how do we optimize the sequence for these 2 therapies? We use immortalized human myoblasts that have a DMD mutation. In this case, we are using a DMD cell that have exon 52 deleted. Therefore, it can be used to test the drug for both 51 and 53 skipping. So the top panel is our 51, bottom panel for exon 53. The left is an experiment that we call RT-PCR to show you that the top band is unskipped mRNA, the bottom band is skipped. As you see, as we use more and more AAV, we can shift from unskipped to a skipped mRNA band. This exon skipping results in dystrophin protein restoration, which on the right panel is a technology called Western blot. It's actually a digital Western blot to show you the dystrophin protein can be restored in a dose-dependent manner for both 51 and 53. So we have licensed a special mouse model, which we call HDMD 3052 MDX mouse model. So this model is a humanized mouse model. So it introduces a full human dystrophin gene into a mouse. However, this full human dystrophin is not wild type. We intentionally remove the exon 52. So therefore, we can directly test a human drug in this mouse model. We don't have to design a mouse-specific drug to test in mouse. So because it's 3052 deletion, we can use the same mouse model to test both exon 53 and 51 skipping. So now what we need to do is to figure out a way to package the U7 small nuclei into AAV. We use a self-complement AAV, and we put triple U7 cassette there. And by using 3 different antisense, we are able to maximize the skipping efficiency. You can see that our antisense, the orange box, is basically binding to either the exon or the exon intron junction. So next, I'm going to show you data of what do we evaluate when we use this AAV vector to treat this mouse model. So first, I'm showing you that the AAV vector can very efficiently go to the heart and skeletal muscle. As an increasing dose from 5E13 to 2E14 GC per kg, we can achieve like 80 copy of AAV genome in heart and 5 to 20 copy in skeletal muscle. Did this AAV vector result small nuclear RNA expression? We are using RT-PCR to show you that the small nuclear RNA can be expressed very robustly, dose-independently, compared to a housekeeping gene TBP. And this amount of small nuclear RNA can result very efficient skipping in heart is 60 to 80%, and in diaphragm and gastrocnemius is 20 to 60%. And this amount of skipping of dystrophin mRNA result in robust dystrophin protein restoration. In gastrocnemius, it's about 10 to 20% restoration for exon 53, for exon 51 this is, and it's very efficient at 60% restoration at heart. And the bottom figure is something we call immunohistochemistry to show you that the dystrophin restored can be properly located in the muscle membrane. You're comparing to the left, actually, is a wild-type animal, and the right is untreated animal data. So is our treatment, can they restore the muscle pathology, right? So we are showing here, again, the left is wild-type, the right is untreated, and the middle is our AAV treatment. The top panel is use serum red to show you the fibrosis in this muscle, and the bottom use myosin heavy chain to show you the muscle degeneration. You can see for both assays, the middle panel AAV treatment is efficiently reduce this muscle pathology. And last but not the least, did our treatment can improve the muscle function? Right, we are using 3 different assays. Number 1 is wire hand to show how a mouse can hand on a metal wire. Number 2 is grip strength, it's showing the muscle force. And number 3 is a treadmill assay as a test of the muscle endurance. For all 3 assays, we can show that this is exon 53 data. We can show that based with our AAV treatment, we can significantly improve this muscle function, force, and endurance compared to untreated control. So we did additional evaluation for our AAV vector for exon skipping. I don't have time to show you the data, but we did show that it not only worked for this particular Delta 52 model, it also worked for multiple exon deletion, which is more prevalent in patients. We also show that this treatment is durable for 6 to 12 months, which is the duration of our mouse study. It also works in a large animal model. We show robust skipping in NHP heart. And we also use bioinformatics to see that there is no measurable detection of off-target. And we can also manufacture this AAV vector in high quality and high quantity. So in conclusion, I hope I provide you data to show that we can use a modified U7 small nuclear RNA and package 3 of that into AAV vector. And after delivered to a mouse model, we can achieve improvement in muscle histopathology, muscle function, and muscle endurance. So we are still in early research for both 53 and 51 skipping. Our future work may include a muscle and heart target AAV capsid, and also a muscle heart specific U7 promoter and enhancer. So I'd like to thank the Regenerative Bio Research and Vector Core team, especially our Chief Scientific Officer, Dr. Olivia Daniels, who discovered the U7 and AAV technology about many years ago. And I'd like to thank Lillian, Cure Duchenne, and all of you for listening. And thank you for keeping us motivated to our research.
Good afternoon. I'm Courtney Young, one of the co-founders and the CEO of Myogene Bio. And today I'm going to be telling you about our work to develop a CRISPR gene editing therapy for Duchenne at Myogene. I wanted to first start by sharing why I got into this field. So below my picture is a picture of my cousin Christopher, who was diagnosed with Duchenne in 2008 at 2.5. And this really brought me into the muscle field. Like many of you, we had never heard of Duchenne before. And I began to dedicate my career to working on muscle diseases. And I've worked in various labs around the world on Duchenne. And I went to UCLA for my PhD, which is actually where we started this gene editing project. So I wanted to tell you a little bit about Myogene, which is a preclinical biotech startup. We're a small group that's now based in San Diego. And as I mentioned, our lead program is a CRISPR gene editing therapy where the first approach is targeting exons 45 to 55, and that's what I'll tell you a little bit about today with some of our preclinical data. And the company was founded by myself, Dr. Melissa Spencer, and Dr. April Pyle, based on work that I had done during my PhD and postdoc in their labs at UCLA, where they're both professors and have worked in the muscle field for many years. We also have additional members of our scientific advisory board, including some that are here. And we also wanted to acknowledge the funding that we've had from the community, including from Cure Duchenne Ventures. So today I'm going to be telling you about gene editing for Duchenne, where I'm specifically going to talk about using CRISPR-Cas9. CRISPR-Cas9 is a relatively new technology. It was actually adapted from bacteria about 11 or 12 years ago. And the idea is that you can use this as a form of molecular scissors on DNA. For example, you can use this in Duchenne models to remove the DNA mutations that cause the disease. And CRISPR has 2 components. One is the Cas protein, which is what you can think of as these molecular scissors. It's actually a protein that will cut DNA. And the other component is called a guide RNA. You can think of this as a flag, and it's basically telling where Cas to cut in the DNA. And although I mentioned CRISPR is relatively new, the first CRISPR therapy, as Dr. Peter Marks mentioned yesterday, was actually approved for sickle cell disease at the end of last year. So we've made great progress with gene editing. This is a little cartoon to show you how the CRISPR-Cas9 system works. As I mentioned, there's two components. That big blue blob is the Cas protein, that's what's cutting the DNA. And then the little red strand is the guide RNA, and that's actually finding the correct site in the DNA through base pairing. So you can specifically design those guides to target specific sequences. That will then bring the Cas protein to that location in the DNA. It causes the DNA to unwind, and when you see that little purple flash, that's the Cas9 protein actually making a cut in the DNA. So the underlying idea with CRISPR is that you can make very specific cuts in DNA at precise locations that we can program in the lab. So we're using this in Duchenne to restore dystrophin. I'm just starting by showing you some of our mouse data where we treat a humanized mouse model of Duchenne that normally would express dystrophin by this green staining, where you see the green staining is ringing all of those muscle cells. That's where dystrophin is normally located in muscle. In our humanized Duchenne model, it lacks dystrophin. You don't see that green staining anymore. But after we've treated with our CRISPR therapy, we're able to restore the dystrophin protein. You can see there's that green staining. It's localized correctly in the muscle cells. So the way this works is by editing out the mutations that would normally cause Duchenne. In many cases, you can think of these Duchenne mutations as disrupting the reading frame, which means that the puzzle pieces of DNA are no longer fitting together, and this is making the gene non-functional. So none of that dystrophin protein can be produced, and that's what is really leading to Duchenne. With our approach, we're using CRISPR, where we're using those flags, those guide RNAs, to target Cas9, those molecular scissors, to cut out exons 45 to 55, which will then put the puzzle pieces of DNA back together again. This makes an in-frame deletion so that an internally deleted dystrophin that's expected to still be functional can be expressed rather than having no dystrophin present. And we targeted this region initially because it encompasses a hotspot. About half of all patient mutations occur within exons 45 to 55. And this is actually a naturally occurring deletion that happens in Becker muscular dystrophy. And Becker patients with an exon 45 to 55 deletion have one of the most mild phenotypes, with some having been described as asymptomatic until their 60s. So we think that we can use CRISPR as a way to turn Duchenne into a mild form of Becker. Some of the advantages of using gene editing is that we're not limited by the carrying capacity of AAV. So our approach is using AAV to deliver CRISPR, not AAV to deliver the dystrophin gene. And that means that we can restore a protein that's much larger. So for example, with our exon 45 to 55 deletion, we make an internally deleted dystrophin protein that's about 87% the size of normal dystrophin, whereas with the gene therapies, they have mini and microdystrophins that are all about a third the size of the normal protein. Because we target a hotspot region, we make a larger deletion encompassing multiple exons. This can encompass 50% of patient mutations rather than a single exon target, which would be 13% or less. And because CRISPR is editing the patient's own DNA, the normal gene regulation, the normal promoter, what drives the expression of the dystrophin protein, is all intact in the patient's DNA. You don't use an artificial promoter to drive expression like with other types of gene therapy. You're keeping the native locus and the normal gene intact, and you're not adding in any new DNA sequences that could potentially cause an immune response. Some of our data in our preclinical studies has looked at evaluating muscle function of these treated mice. This is looking at a wire mesh hang time test. We take the mice, they grip a wire mesh with all 4 limbs, we measure how long they can hold on. And in gray, the untreated mice aren't able to hold on for very long, but after we've treated them, their hang time has improved to a level that's basically equivalent to the healthy mice. And then we also measure in an open field test how far they move after we've exercised them. And again, compared to the untreated mice in gray, after treatment, the mice are able to move a larger distance similar to the healthy mice. So we're currently still in preclinical development with this gene editing therapy. The idea is that it could change the patient's own DNA, aiming to create a larger, more functional dystrophin while still targeting a larger proportion of patient mutations than single exon targets. We've shown efficacy in human patient-derived cells as well as this humanized mouse model. And our upcoming studies include testing in a large animal model efficacy study, conducting very thorough safety assessments, as well as manufacturing to begin a clinical trial, which is potentially happening in the next few years. So in summary, Myogene is a startup that's been really inspired by patients to develop truly meaningful therapies for muscle diseases. We think that CRISPR gene editing is kind of the next generation of gene therapy, where you can use it to permanently remove patient mutations. And the idea of this initial target around exons 45 to 55 can be expanded into other regions of the gene and even other muscle diseases to really build a platform of approaches to target a variety of patient mutations. And we're interested in using this to grow Myogene as a muscle disease therapeutics company, and we're currently aiming to continue to raise money to complete development of this and additional therapies. And thank you all with your attention, and please feel free to contact me.
Good afternoon, everyone. My name is Sherif Bordbar, and I'm a co-founder and CSO of Therapeutic. First, I wanted to start with the fact that it's personally very special for me to be here. I also grew up in a family with muscle disease. My father has FSHD, and that was what motivated me to start working in this field many years ago. I remember about 15 years ago, I was checking the websites of Duchenne Muscular Dystrophy Association, hoping one day I could contribute, and I'm very excited to be here. Therapeutics is a muscle gene therapy company developing next generation gene therapies to address the main issues of the first generation therapies, both in terms of viral delivery and also development of new regulatory elements. Today I'm going to talk about the work that we've done to develop KT-809, which is our gene therapy development candidate for DMD. It's a very exciting time for muscle gene therapy. There are multiple different first-generation gene therapies in development. However, there are headwinds. There are shortcomings with the first-generation gene therapies. The naturally occurring capsids, AV8, AV9, AVR74, used in these gene therapies are mainly targeted to the liver. They require injection of extremely high doses of virus to be effective in muscle, and using high doses resulting in liver toxicity, complement activation, potential kidney injury. So there is certainly a huge unmet medical need to develop more effective viral gene delivery vehicles to deliver genes into muscle. Another issue that I'm going to touch on is that the first generation gene therapies don't necessarily have highly optimized regulatory elements that can result in high levels of expression of transgene in a skeletal muscle, specifically in heart in non-human primates or both tissues. So we're combining two different platform technologies at Kate. One of them is a capsid engineering technology. We use a technology named directed evolution and combine it with machine learning to basically engineer the surface of these capsids to enable them to effectively go to the tissues that we're interested in. And in this case, it's the skeletal muscle and cardiac muscle. We also are developing high throughput, we're using high throughput approaches to develop enhancer or promoter regulatory elements that enable us to express the transgene in the desired cell types for our indications of interest. For Duchenne, it's going to be both skeletal muscle and heart in large animal models. This is a schematic of our capsid engineering platform. We prepare millions of different variants of these capsids and then do our directed evolution mainly in non-human primates. And the reason for that is the transduction of AAVs have been shown to be different across the species. So the transduction efficiency that you see in mice doesn't necessarily translate to primates, to larger animal models. And we perform most of our experiments in monkeys, in non-human primates, to make sure that it's translatable to the clinic when we start our clinical trials. we have used this technology to evolve a new family of capsids that we call the myo-AAV class of capsids. And as you can see here, they result in much higher expression of the transgene, which is here a green protein named GFP, green fluorescent protein, in skeletal muscle and heart, in this case, 57 black 6 mice. And now we have taken it to the next step and performed the experiments in non-human primates. I'm not going to spend a lot of time on this slide. This is just to show that we have used this technology previously to develop a candidate for another genetic muscle disease, X-linked myotubular myopathy, which did have a gene therapy in the clinic using extremely high dose of virus. Unfortunately, there were serious adverse effects and patient deaths in some of the patients that received that experimental gene therapy. So there was a clinical hold, and the company that was moving that product forward, Astellas, have now licensed this new candidate for X-linked MTM gene therapy that we developed at Kate, and is moving that forward towards clinical trials. So there will be hopefully clinical data for the myo-AAV class of capsids pretty soon. This is showing the new generation of myo-AAV class of capsids that we have evolved that not only transduce the skeletal muscle and heart. So the green signal shows that it's expressing the transgene in skeletal muscle and heart. You see multiple different skeletal muscles, heart and liver in this slide. When it says vehicle, it's a control, so it hasn't been injected with anything, or just a buffer, just a saline. And you don't see any expression of the green protein there. myo-AAV 3.8 is a first generation capsid that we have used previously, and it transduces skeletal muscle and heart very effectively. It also goes into the liver. myo-AAV LD 1.2 is a new generation of these capsids that we evolved to be also actively detargeted from the liver. So to give us higher safety margin, to make sure that we can not only transduce the skeletal muscle and heart very effectively, but also don't get into the liver. We have now developed a 6th generation myo-AAV capsid that is extremely potent in transducing skeletal muscle and heart in non-human primates. Here you can see the comparison of myo-AAV LD 6.1 to multiple other capsids currently in development. And you see, compared to AAVrh74, which is used in AAV-approved gene therapy, myo-AAV LD 6.1 can transduce non-human primate monkey skeletal muscle about 60 times more efficiently, about 20 times more effectively in the heart. At the same time, it results in 10 times lower liver exposure. So much higher expression of the transgene in the target tissue, much lower exposure to liver. The liver is part of the equation. We're also developing regulatory elements that can transduce, that can basically express the transgene, which is here a microdystrophin transgene, in both skeletal muscle and heart in large animal models. And here is the result of a selection that we have done in a systematic high throughput manner. So we tested thousands of different regulatory elements that could potentially result in high activity in skeletal muscle or heart directly in cynomacaques, non-human primates. Here, y-axis is the level of expression that you see in the heart. The x-axis is the level of expression that you see in skeletal muscle. MHCK7 is a promoter that's used in FDA-approved gene therapy. And you can see, although highly active in the skeletal muscle in non-human primates, is not super high in the heart. And we have now identified new regulatory elements that we're using in KT-809 that result in similar or higher level of activity in skeletal muscle, but definitely significantly higher activity in the heart. So I'm going to talk about KT-809 for the next couple of minutes now. It uses our top muscle-tropic liver-detargeted capsid, myo-AAV LD 6.1, a novel enhancer promoter element that we identified in non-human primates to be active in both skeletal muscle and heart, and a codon-optimized microdystrophin transgene. We have performed experiments in both dystrophic animals, MDX mice, and also in non-human primates that I'm going to show you the data for. And we have compared it to an FDA-approved DMD gene therapy, which basically is AVRE74 MHCK7 microdystrophin. It's a surrogate of that FDA-approved gene therapy because it contains a flag tag or a small portion of the protein at the end so that we can show where it's expressed on the surface of the skeletal muscle fibers and cardiomyocytes in non-human primates. The first experiments are the comparison in MDX mice, and you can see here that we also used the surrogate of the FDA-approved gene therapy at the clinical dose. It's the dose of 1.33 to the 14. The dose that we're using for KT-809 here is the dose of 4e to the 13, so 3 times lower dose. And at the dose of 4e to the 13, we're getting transduction of 100% of the skeletal muscle fibers in MDX mice. When we do the comparison compared to endogenous human dystrophin in skeletal muscles, we're achieving about 80% of normal human expression. In contrast, the surrogate of the FDA-approved gene therapy is resulting in about 10% of normal human dystrophin expression. This is the quadriceps muscle. In the mouse heart, the results was interesting. So both of these compounds resulted in transduction of 100% of cardiomyocytes in mice. And we had about 3 times higher expression from KT-809 compared to the surrogate of the FDA-approved gene therapy at the clinical dose. However, the results were very different in non-human primates. We also performed functional analysis of the muscle to show that if you have enough of the microdystrophin transgene, are you going to improve the function of the muscle? So on the left, you see that the wild-type animals in orange are stronger. Their muscles are stronger compared to MDX animals, which is mouse model of the disease. The animals that received KT-809 in green have almost basically complete reversal and similar amount of force compared to wild-type animals. And the animals that received the surrogate of the FDA-approved gene therapy has an increase in force, but not to the same level. If you look at the bottom right part of this slide, you see the comparison of how much protein is expressed in these muscles. And you can clearly see that the more microdystrophin protein you have in the muscle, it results in higher force of the muscle. So if you have enough of microdystrophin expression, it should improve multiple different cycles. The force of the muscle decreases in the dystrophic muscle, which is shown in blue here. And then the wild-type animals are mainly protected. Those are in orange. The animals that received the surrogate of, but then the animals that received KT-809 at 4E13 have about 80% of normal protection against damage. Again, another indication that the levels of microdystrophin expression and the uniform expression of microdystrophin is extremely important to improve the function of the muscle. We did the same experiments in non-human primates, in monkeys, basically used the same virus that we used in the mouse experiment, and this is what we found in the heart of non-human primates. So this has been an open question of how gene therapies that are currently in development are expressing the transgene in the heart. The red signal here is how much microdystrophin you get in a monkey, and you see that animals that received KT-809 at a dose of 40 to 13, we see about 80 to 90% of cardiomyocytes in the monkey heart to be expressing microdystrophin. We could barely detect microdystrophin expression after injection of a surrogate of an FDA-approved gene therapy at the clinical dose. We also quantified the total amount of that microdystrophin, and we had about 60% of normal in the left ventricle of those animals. This is a skeletal muscle from the same animal. So there is some level of expression in the gastrocnemius from the surrogate of the FDA-approved gene therapy. We are able to detect 100% of skeletal muscle fibers to be expressing microdystrophin and about 50 to 60% of normal human dystrophin in the muscles of animals that receive KT-809 at a dose of 40 to 13. This is pectoralis major. In this muscle, we could achieve about 100% of normal microdystrophin. Every single muscle fiber in every muscle that we looked at in these animals were expressing microdystrophin. And what made us really excited was the diaphragm, that we were achieving about 100% of normal expression in the diaphragm. All the muscle fibers were expressing dystrophin. At the same time, when we look at the liver of these animals, we have much lower exposure to the liver. So Animals that receive KT-809 at a dose of 4e13, they have 27 times less number of vector genomes per diploid genome in their livers compared to animals that receive the surrogate of FDA-approved gene therapy at the clinical dose of 1.3 × 10^14. So clearly shows that we can express close to physiological levels of human dystrophin expression in both skeletal muscle and heart in non-human primates with KT-809, and more importantly, uniform expression across the muscle. And we have about 27 times lower liver exposure, which gives us much higher safety margin for this specific compound. At the end, I would like to thank the Therapeutic teams for basically working very hard to generating this data, and thanks for giving us the opportunity to present the work.
That concludes our session on new therapeutic approaches. If we can just have another round of applause for all of our speakers today. Thank you so much. Thank you. And we're going to be getting ready for the next session, which is clinical trials and clinical trial design, lessons learned and implications for the future. But I think we need, are we all set, or do we need to still be miking people up?
Still looking for Susan. There she is.
Sue,
Come on up.
follow me out there. I could see her.
Well, good afternoon, everyone, and it's a pleasure to be up on stage with Barry and Sue, who will be up very shortly. I want to again thank the Miller family for putting on such a great conference.